Okay, so in the last segment, we had determined that the Z gene is in the middle of the sequence of these three. And so we've rewritten our parental chromosomes this way. And now we're going to try to figure out the distance between X to Z and Z to Y. So in order to do that, we need to know what would a crossover be? What would a crossover allele combination be if a crossover occurred in segment one? So we're defining segment one as this. So if there was a crossover, you'd get an X with a little z, little x with little z, and a big X with a big Z. Those would be the allele combinations for a crossover in segment one. And you remember that genetic distance is equal to recombination frequency. So we just need to know how many recombinations, how many crossover offspring had a crossover in segment one. So we write down these two genotypes here. These are the ones we're looking for in the data. So I got to go back to the data, and I'm looking for a little x with a little z. And in order to do this, you need to ignore the y. So actually, I think I did it here. Okay, you need to ignore the y. So here I put a big old block over the y. Now look at the y. The y is not in segment one. So the question is, little x and little z, that's going to be a group that has a crossover in segment one. Little x and little z, that one has a crossover in segment one. And then big X and big Z. And we're ignoring Y because Y isn't even in segment one. So we should find 4 groups, and we're going to add those all together and then divide by the total. And that gives us the recombination frequency, RF, and it's 17.5% in this problem. And so that's going to be 17.5 map units for segment one. Now for segment two, we need to do the same process. And what you're going to do is for segment two, let's go back to our parental. We're going to look for a big Z with a big Y and a little z with a little y. All right. And I'm going to go back to the original data set here. So again, this time we're ignoring X. So big Z with a big Y and little z with a little y. And there should be 4. So this time it's going to be 13, 95, 90, and 15. And we'll add those together and divide by 1,000. And that one comes up to be, I think, 21.5 map units. And the final map looks, 21.3, sorry. The final map looks like this. So Z is in the middle. It doesn't matter if you put a big Z or a little z because different organisms have different alleles at that location. But the distance between some allele of X and some allele of Z is 17.5 map units or centimorgans. The distance between some allele of Z and some allele of Y is 21.3 centimorgans. It would not matter if you wanted to put Y over here and X over here and Z in the middle, but you need to make sure that this is the 21.3 in that case, and this is the 17.5. So that would just represent a rotation of the chromosome, and that would be fine as well. Something else you might be asked, what is the arrangement or configuration of alleles in the mother? Because you remember that the mother is the triple heterozygote individual. This is going to be a word answer or a sentence answer. You're either going to say that the alleles are in the cis arrangement. Sometimes the word coupling is used, but I use the word cis. That's just the way I was trained. And then, or trans or repulsion arrangement. I'm going to say trans because that's how I was trained. What this means is you're looking at, in the mother, right? Remember, the mother was little x, big Z, little y on one chromosome, and big X, little z, big Y on the other chromosome. You're looking for the arrangement of the recessive alleles. You're only looking at the recessives. So in words, the way you would explain this layout that I've drawn here is the X and the Y are on the same chromosome, and the Z is on the other one, on the homolog. And so we would say X and Y are cis. Cis means on the same chromosome, and Z is trans to both of them. X and Y are in a cis configuration, or you can just say X and Y are in cis, and Z is in a trans configuration or trans arrangement. Okay, so it's a sentence, and you're basically explaining in words this layout that we've drawn here at the bottom. All right, so that is the answer. And you really need the whole, you need the whole sentence. X and Y are in cis, both in trans to Z. Or Z is in trans to both of them, either way. Other calculations you'll be asked to do. These are equations you should either memorize or, memorize, I think is the only way. Is you're going to have to predict the expected number of double crossovers. So we observed a certain amount of double crossovers in this last problem. We observed, 13 and 15, so a total number of 28. And what we're wondering is if the map, based on the map that we came up with, how many double crossovers would we expect based on the size of those segments? And so what we do is we use the recombination frequency for segment 1. So I would use the decimal version, because if you do 17.5%, but you forget to hit the percent key on your calculator, then you'll end up with some kind of enormous number as your answer. And you'll know it's wrong, but you might not know why. So just use the decimal version, 0.175. And then recombination frequency for segment 2, and then the total. So what this is if the recombination frequency, the frequency of crossover in segment 1 is 17.5%, meaning 17.5% of meiosis have a crossover in that region. If that's what we measured in the past in data we've already observed, we would predict that rate would continue into the future. So the recombination frequency becomes the recombination probability in the future. And so we're saying if there's a 17.5% chance of a crossover in segment 1 and a 21.3% chance of a crossover in segment 2, then the chance of a double crossover would be the product of those separate events, the product of the probabilities of those separate events multiplied by the total. So in this case, we end up with, I think I put the numbers on the next page, 37.275 is what that comes out to be. And then you're going to be asked to compare that to what it has been observed. So 28 were observed, 37.5, is that what I said? Yeah, 37.275, sorry, is what was expected, and you make a ratio or fraction of that. And that's called the coefficient of coincidence. How well does your observed and your expected match up? If the coefficient of coincidence is 1, that means you observed exactly what you expected. That's pretty unlikely, but it could happen. In this case, 28 divided by 37.275 is 0.7512. And so you see that calculation there. And then the last bit is called interference. Interference is sort of the flip side of coefficient of coincidence. You take the coefficient of coincidence and subtract it from 1. So if the coefficient of coincidence is 75.12%, then the interference level is 24.88%. And it's just a measure of the amount of double crossovers that were somehow interfered with or inhibited or repressed. Usually we think that it's a steric problem. Steric meaning a problem with the amount of space there is, because you have small map segments. The enzymes needed to do a crossover are large proteins. So if one recombinase enzyme comes into segment 1, do a crossover, even if another one is trying to come into segment 2, it might be sort of pushed away or blocked from being able to get access to it. So we call that interference. So if interference is 0, that means your coefficient of coincidence was equal to 1. And like I said, that means that you got exactly the same amount of DCOs observed as what you expected. If interference equals 1, that means your coefficient of coincidence was 0. That means you didn't get any DCOs at all. So they were all inhibited. They were all interfered. It's very unlikely to get interference of 0 or interference of 1, but those are theoretical boundaries on what the value should be. So in this case, ours was close to 0.25. It means that a fourth of the DCOs that we should have expected in our data were not observed because something interfered with them forming.